On behalf of INCT, we are very happy to have here today Dr. Praveen Arani. He's a dentist, scientist, and academic known for his work in neurobiology and regenerative medicine. He's a professor at University at Buffalo School of Dental Medicine, where he conducts research focused on tissue repair, wound healing, and therapeutic applications of light, especially in the field known photobiomodulation. So Dr. Arani is also recognized internationally for collaborating with different researchers and institutions, helping everybody around the world to work with lasers. He's a bridge from basic science and clinical application in healthcare. So Praveen, for us, is a huge honor to have you as a friend, as a colleague that work with laser and with us in this INCT. Thank you, and the floor is yours.
Thank you very much, Manuela. I really appreciate meeting all my colleagues, and thank you for the very kind words. I was asking just before we started, I was trying to share my screen, that the audience is probably a mix of people who are experts, clearly. I see Eleni, I see Dr. Martins, Dr. Figueroa. I think all of you have already done photobiomodulation way more, with expertise than I can show you. But I do have some slides, assuming there are some students and postdocs and trainees who would actually benefit from some basic fundamentals. Can you see my slides?
Yes.
Okay, excellent. And again, I would be more than happy to share my slides because I'm going to go through some of the material pretty quickly. I'm happy to send the slides over to Manuela, and please feel free to send it to everybody. That is absolutely what we want to do. All right. So with those words, let me begin. So we just published a new paper on mechanisms of photobiomodulation, which I thought was very exciting because all of us who work in the field are trying to understand how to bring all the therapeutic potential to a larger audience. And one of the things that I think all of us agree, one is obviously dosimetry and how we are giving the light treatment. The other part is how is this thing working in so many different diseases. And that is exciting, but it's also, if you have ever talked to a colleague who doesn't know this treatment, they always look at you as if, you have two horns and a tail and extra, limbs or something, because they're like, how can this work in so many different diseases? So I think now that we are getting a better understanding of different mechanisms in different disease contexts, it is beginning to make some sense. And I hope today I can show you some of the progress we have made. Obviously, a long way from solving anything, but hopefully in the right direction. So the title of my talk is Confluence of PBM Mechanisms. And for everyone here, I'm sure you love the mitochondrial mechanism and cytochrome C oxidase. But hopefully you've also heard about some of the other mechanisms. And it turns out, as you would expect, everything works well in collaborations and in teams, and so do the mechanisms. So you will see, hopefully, I'll spend more time on that part, the new paper, but I wanted to give some introductory comments and some background so we can move towards that. So let me begin. These are my disclosures. I work with several companies and institutes, and we are very active with very many institutes, several colleagues leading from Brazil, Dr. Alan Silva, Dr. Fabio. There are so many people who are leading the field who are also part of these organizations. So let me begin by one of the most common questions we get asked. Light treatment has been around for the longest time. What is different about photobiomodulation compared to some of the better known cousins, right? Phototherapy, lasers, photodynamic therapy are better known than photobiomodulation in many clinical and medical contexts. So one of the things that I always like to start my talk with, and this is the actual ADA Technical Bulletin 189, and I will show you that update in one of my later slides. We know that when you're using light for surgical treatments, you're primarily using the heat generation to ablate or evaporate, right? If you use it at slightly low levels, you can also vasodilate or vasoconstrict or coagulate, depending on how you're using them. Photodynamic therapy and photobiomodulation are often confused as things that are similar, but obviously they're not the same. A major difference between photobiomodulation and PDT is that photobiomodulation is non-thermal, non-destructive, and it can modulate biological responses, inhibit, stimulate, or change the immune response. However, technically, it should not destroy the target. Destruction of the target is a photodynamic action. And if you're doing that in many different clinical contexts, that the difference is that PDT is destructive. So this is one way that we have been able to distinguish. Both are very, very helpful. PDT is amazing, but it is not the same as PBM. And in many contexts, especially peri-implantitis, people have started combining PDT and PBM and have getting wonderful results. So this is one of the distinctions that I'd like to make. I mentioned several significant milestones, everything from the terminology from over 350 different terms, like low-level light therapy, cold laser therapy, infrared therapy. I think all of that. And of course, the most popular in social media, everyone from the Kardashians to Ronaldo is actually using red light therapy, right? So all of these are now formally, technically called photobiomodulation. And this is now a PubMed mesh term, which is how the entire scientific literature is indexed. In 2018, we also were fortunate to have photobiomodulation as a part of the US healthcare public law. And many people don't know this, but besides trans-electrical neural stimulation, photobiomodulation is now federally mandated to be explored as a pain relief mechanism. We know this, it is very effective, and there's a lot of exciting new research with blue light that I keep getting asked about. There's a lot of interesting research and clinical evidence for its pain relieving properties. And therefore, I think it's going to get a lot of attention. Several of the leading Brazilian institutes, especially Dr. Antunes, several Dr. Silva, so many people have contributed to these oral mucositis protocols that actually have become now standard of care. So photobiomodulation now is standard of care for mucositis. I'm telling you, and you're telling us. You've been telling us for a long time, but I think it is now formalized that it is actually standard of care, right? Following that acceptance by both the MASC, ISO, and the WALT, WALT, FDA finally recognized photobiomodulation as a discrete form of treatment, and it is now recognized as a separate kind of treatment. And this has led to several, I'll show you in a second, several approvals of photobiomodulation devices, which are meant for photobiomodulation. They are not sauna devices, they are not phototherapy devices, and they are not PDT devices. This also led to the ADA recognition along with lasers in dentistry, which is Technical Bulletin 133, and 189, which is this particular technical bulletin. This has recognized photobiomodulation as a discrete form of light therapy by the American Dental Association. So as soon as it was recognized, we were very fortunate to immediately get insurance codes, right? CDT codes. So as of January 1st of this year, photobiomodulation now can be performed formally and can be reimbursed. So that's a huge milestone for our field, the recognition and the availability of photobiomodulation. So I mentioned some of the FDA approvals. You probably have seen these already for dry age-related macular degeneration, for fibromyalgia, and earlier this year for mucositis. This particular company actually got approval for oral mucositis with an intraoral device, which is shown here. So there are several FDA-approved devices, not just FDA-cleared, but FDA-approved devices for specific pathologies. And if you're working in any of these areas, this is a huge, huge, huge availability, at least in the US. It is the availability of these devices for specific diseases. Again, I mentioned this studies that have been done. And again, several leaders in the field who are, I'm sure, a big part of this forum as well, have actually shown that photobiomodulation should be provided to every cancer patient. right? Every cancer patient, before they get the cancer treatment, irrespective of the type of the tumor, where the tumor is present, the treatment complications can be mitigated or reduced in severity. You may not be able to prevent some of the complication, but you will reduce the severity of many of them. And again, all of you have, in one way or the other, I think, contributed to many of the studies. So that is really helpful. So I don't know how we're going to take questions. I'm happy to stop at any point and take questions either in the chat, or if you can just, unmute yourself and ask, that's fine too. I have divided my talk in several sections. And before I go to the technical part, please feel free to stop me, and I'm more than happy to discuss it. So then, if we all know that PBM is so great, why aren't more people using it more routinely? And again, you and I think we have discussed this in many different forums, but this was one of the summaries that we published where we asked the question, why is PBM not more consistent? And again, things like, differences in our patients, differences in the dosing, whether it is provided in a particular way or the device parameters themselves. And many people are now combining different wavelengths. And as we understand more and more of the mechanisms, it is becoming evident that the non absorption wavelengths can also have a very potent effect, especially 810 nanometers, 940, 980. We know that these non absorption wavelengths also have very potent clinical benefits. So this has been studied in great detail by many labs, and these are some of the concepts that we have actually done in my lab. And it basically boils down to this particular image here, where we have begun to think about photobiomodulation as both an absorption, but also inelastic scattering. And that makes the other non-absorption wavelengths more understandable. How do these things work if there is no light absorption? Is because energy can be transferred by inelastic scattering. And that is the whole process of the energy transfer phenomenon. Now, it is not just enough to transfer energy, but it is also important to transform energy. And when you're transforming the energy within the biological system, we know that heat has a minimal role, if any, to play. And I'll show you some data behind that. But we do know that redox changes are absolutely critical, right? So we know that ROS generation, as well as change in conformation of different biomolecules, especially proteins, is particularly important. So there are these transformation events that make a physical form of energy, light, into a biological effector response. So we understand this, and this has helped us outline, again, this is a paper that Dr. Silva's group put out. We collaborated on this. So we were able to show that if you use very standard parameters to communicate your protocol, we can do it in a more uniform way. And because the light, once it enters, a photon enters the tissue, it becomes much more difficult to understand what it's doing inside the tissue. But what we can absolutely measure with lots of confidence is what happens on the surface when the light hits the surface. And that is the concept of tissue surface irradiance. As we understand the process of photobiomodulation, we are beginning to understand that even the wavelength of each photon, the energy of the wavelength of each photon, is actually very, very important. And therefore, we want to ensure that we account for that. So rather than just doing watts per centimeter square into time, which we all standardly do for our fluence, we have started adding the photon energy. And this whole concept is called a photon fluence, not just fluence, but a photon fluence. And to ensure that all of us communicate in the same language and the protocols, we have started using 810 nanometer at 3 joules as a standard for us, so that everybody who's doing with any wavelength can convert it into an Einstein number, which is basically divide that number by 4.5. And this paper actually goes step by step explaining how this process is done. And again, if anybody's interested, I'm more than happy to get on a call with you and discuss the concept much more in detail. So the other thing besides the delivery has been what is the mechanism, right? And to explain this concept, I find that giving an example of a wound like this, right? This is a chronic wound on someone's leg. And what do you do in this process, right? If you have a chronic wound like this, The first step is to basically debride that wound, right? And when you're debriding the wound, you would use like a sharp curette or a blade to debride all the dead and the necrotic tissue, and then do the disinfection and then put a bandage, right? When you debride a wound, there is a normal cascade of healing that occurs. One of the most common mistakes or confusions I find in the field that we have, discussed with many of our colleagues, is that you ascribe some of these changes to the mechanism, right? And that is obviously not true. The blade does not induce VEGF in the blood vessel or PDGF in the fibroblast, uh, or TGF-beta, which I'm going to show you in a second. It is not the blade or the curette or the scissor that's actually doing it. It is the normal process of changes. And this is what we would think of as the secondary or effector responses. So all of these that occur as a natural process are not true mechanisms. They are just processes which have been started because of the healing or the physiological change. A true mechanism, which is a primary causal event, is induced by the intervention, right? So when we talk about light absorption or scattering, when it changes a biological target, that is a mechanism. When you talk about cytochrome C oxidase, it has to absorb the light to increase the amount of ATP and ROS, right? It is not the ATP and ROS which are mechanisms, it is the absorption of light by the cytochrome C oxidase. So there is a distinction between a primary causal event and a secondary effector event. And we have learned in the last few years a lot about what happens with photobiomodulation. We know we can modulate pain, inflammation, and immune. We can either increase it or decrease it depending on what we are trying to treat. And we do know we can stimulate healing and regeneration. And all of you are very familiar with all of these mechanisms, right? So we have cytochrome, or some of my things are not showing up, but we all know that mitochondrial cytochrome C oxidase is capable inside the cell of absorbing light. We know that there are several receptors and transporters on the membrane which can modulate either neural conduction or other kinds of activated signaling. And outside the cell, we have certain molecules like the TGF-beta molecule, which can be activated by light. So there are different areas, and you know that light goes everywhere. When you shine light on a patient or even a cell culture dish, it is going to go everywhere. So it's going to hit multiple targets at the same time. And that is the new paper that I will talk to you about in a second. And again, I will send you my entire presentation if you're interested. These are the references for those mechanisms, which all of us actually know pretty well. And these are the other references for the membrane receptors. And this one is the specific work that we do in our lab, where we have shown that if you activate redox, ROS, in low amounts, you can change several proteins, and many of the proteins have been not characterized. So there's a huge area for the students and residents in the group who are interested in doing more research. It's a blue sea out there. It's a blue sky research out there. So there are lots of proteins of interest that you can actually, proteins, nucleic acids, lipids of interest. One of these proteins turns out to be TGF-beta, the molecule that we are very interested in. And you probably have seen this in some of my other talks, where we actually show that a specific mutation on the protein structure makes the entire complex redox insensitive. And when you make it redox insensitive, a single amino acid, a methionine substitution on the entire protein, none of the others, but one single amino acid on the protein, when you mutate it, you can make the entire complex redox insensitive. And that prevents activation by photobiomodulation. So this is the regular photobiomodulation wild type cells. You can see that it activates the signaling via SMAD2, but this does not happen when you have that mutation. So clearly showing us that photobiomodulation, among many of the mechanisms, we have gotten it down to a single amino acid in one particular case. And there are, of course, many groups that are doing this in many other contexts. And again, I don't need to tell you this, but we have done human clinical trials where we have taken biopsies from tissues, and we see that immediately post photobiomodulation, you can see that there is increased TGF-beta immunostaining, showing us that the TGF-beta mechanism is also operative in vivo in humans and also in animals. So we have summarized all of that in this particular review, where we talk about photobiomodulation in several cancers, uh, cardiovascular disease, as well as aging. And all of these mechanisms are, I think, very, very relevant to how we would do clinical photobiomodulation. So, are there any questions so far that I may answer about just the background, or I can jump into some of the research now?
I think
I
you
don't see
can.
any.
I think that you can still go in, and at the end, maybe we can have
Sure.
some questions. But it's
All right.
amazing. Yeah.
Sounds good. Sounds good. So when we think about photobiomodulation dosing, one of the most common questions we get is that what is changing, right? How do you know how much dose to give and how do you stop giving dosage? And this is where I think the key question about the Arnschulz curve, which all of us know very well, and we think about all the time. We think about why you would give the right amount of dose at the right time and the right place, and how many times should we repeat the treatment so that you're giving enough dosage for the benefit of the patient. Now, one of the things that I'm sure you might have heard in other forums as well is that photobiomodulation does not cause harm. It does not cause damage. However, if you give too much dosing, you will negate your benefits. You will neutralize the benefits you're giving. And this one came from several studies in the lab, but the most important study, which I'll show you in this particular graph, is that we have used different kinds of imaging and sensing mechanisms like an infrared camera, an ultrasound Doppler, as well as other photoacoustic imaging systems. And we are trying to do this is Monica and Elisa in the lab who are doing these studies on animals and with anesthesia and ethical approval. And what we are trying to do is understand how much dose is too much dose. And they found that when you do a specific amount of energy deposition, as low as elevating the temperature to 45 degrees, what happens in these systems is that you get, where is the, oh, there's a background is changed, but you get elevation of temperature between 42 and 45 degrees. At that temperature, 42 to 45 degrees is not even warm coffee, right? So, our coffee is hotter than 45 degrees. But at this temperature, many of the enzymes that neutralize ROS are inactivated. And one of the enzymes that we have studied in some detail is catalase. So when you're doing photobiomodulation treatment and you elevate the temperature, at 45 degrees, you get neutralization of the redox scavenging effect. Therefore, at this point in the treatment, the damage threshold becomes vertical. So what you should see here is a line going this way and a line going up, right? And suddenly you start seeing subcellular damage. You don't see it at the clinical level, but you're doing this at the molecular level. So there is significant damage, which we have validated in the animals. Again, and this is the work that we focused on this molecule called ATF4, activating transcription factor 4. This turns out to be a master regulator of when the damage makes a decision on the cells to survive versus commit to a process of apoptosis and autophagy. And all of this was summarized in a review that I'll show you in a second, this one, Translation Medicinal Review. And we have validated these mechanisms in vivo. Now, you can ask the question, can we neutralize the benefits or neutralize the damage or improve the benefits by changing the thermal threshold? And in this particular study, we did that exact study where we actually cooled the skin and started doing more and more doses of photobiomodulation. Interestingly enough, you can completely neutralize the damage by doing this process, but when you neutralize the redox amount, you don't get complete rescue, showing us that the heat response is the more important response in terms of the upper dose threshold. It is not the redox by itself, but it's the heat generation. And that's why the ROS scavengers are really, really important. So we have, oh, this is the thing I was going to show you. So this is the actual escalation of the damage threshold. And you can see that at this point, because you're neutralizing the redox, you start seeing all of the damage. And this has been summarized in this picture here. So you can see this image here on the right side. I hope you can see my mouse, where you can see that this molecule in the endoplasmic reticulum is coordinating all of the molecular signaling and is responsible for the heat shock response integrated stress response, and the decision of the cell to either do apoptosis or autophagy. So all of these things that are happening inside the cell, which we obviously cannot see in real time, are determining your therapeutic threshold of how and when you do photobiomodulation and how much energy is optimal to do maximal benefit for photobiomodulation. Now, this is just the summary again. And many of the studies, follow-on studies, including several clinical trials, are focused on validating this in human treatment disease models. So that's what we use as a clinical and the molecular biomarker. And we all know that photobiomodulation has so many different applications, right? And one of the most interesting things in my lab is we focus on dentine and odontoblast biology. And you can see that we know that TGF-beta and has a central role in both in stem cell fate as well as in dentine homeostasis. And I don't know if you have seen this paper before, but we have focused our interests on driving odontoblastic differentiation from dental stem cells to odontoblasts. And we know that this contributes to osteodentine or tertiary dentine. We have also done similar experiments in third degree burn wounds. And in this particular case, we use a transgenic animal chimeric knock-in model. When you use the regular wild type littermates and do third degree burns, you can see all of us have done different versions of these experiments. We can see that photobiomodulation is able to improve the healing response. But when we make a chimeric knock-in, where we have put in a different isoform inside the latency-associated peptide, you can see that it does not get activated. I'm sorry. And because you cannot activate the latent TGF-beta complex, you can see that you do not get the benefit. So at least in terms of tissue healing and regeneration, we know that the TGF-beta mechanism is central to the ability of PBM to produce therapeutic benefits in the patient and in the in vivo context. So I'm sure there are many mechanisms, which is the paper I'm leading to, but we know that TGF-beta mechanism is actually really important. We have also done several studies in peri-implantitis, and we find that when you look at what does light-activated TGF-beta do, we found that it can induce a very, very potent antimicrobial peptide called beta-defensin 2. This beta-defensin 2, as we know, is going to reduce the biofilm and improve the host healing response, but it does not directly destroy the biofilm. So when you do photobiomodulation in peri-implantitis, you must begin with scaling and root planing and disinfection. Only then is photobiomodulation going to be helpful. And we did a clinical trial, and the results will be published later this year, showing us that this is actually relevant in vivo. A very talented student from Brazil, again, who came to our lab, asked the question, what happens to the blood vessels when you do photobiomodulation? And in a very elegant set of experiments, Victoria actually showed that when you do photobiomodulation and activate a specific pathway, that pathway in that cell type will also activate other pathways. And for her, she specifically focused on VEGF, a vascular endothelial growth factor, as well as PDGF. And she found that depending on when the vessel, the endothelial cell, is actually going to migrate, first proliferate, migrate, and then form the blood vessels, it activates different signaling, all beginning with the photobiomodulation response, which makes sense. So I was talking about the secondary effector responses. There is a lot of coordination between different pathways to get to the therapeutic response. In another very elegant work by, again, inspired by what they're doing in the clinic, Amanda, who's another student who came to our lab from Brazil again, she actually very nicely showed that in the venom bite patients that she was actually treating with photobiomodulation, she was asking the question, which is my optimal wavelength, and how do I know which cell type it's going to actually modulate? So she focused her analysis on macrophages. And surely enough, as you can see in this particular, you can read the paper here, we found that unlike endothelial cells, fibroblasts, odontoblasts, keratinocytes, macrophages require a different type of treatment. And that's what we have actually shown in this paper here, both in terms of wavelength as well in other papers we have shown dosing differences for different responses. So and we know that cancer cells, which we often get asked all the time, what happens to cancer cells in the field of photobiomodulation? And I will point to the two clinical papers and a very elegant, again, in vitro paper, and several of our work, some of it is still unpublished, where we showed that the tumor cell will respond to photobiomodulation energy in a very different way than a normal cell. In fact, the tumor cell undergoes apoptosis and autophagy, while the normal cells will proliferate, migrate, and differentiate. So clearly, there is a difference in the cellular response to the same energy. You're doing photobiomodulation on your patients. Every cell is getting a different amount of energy, right? And here's an example of that. So in this paper by Carl Engel, we showed that epithelial cells, keratinocytes, will respond very differently than fibroblasts, right? So what I'm trying to tell you is that when you do photobiomodulation treatment, you're going to first activate multiple mechanisms. You are also targeting multiple cell types. There is no way to only shine light on an epithelial cell or a keratinocyte or an endothelial cell. In the patient, you're going to end up, or even in an animal, you're going to end up irradiating multiple cell types, right? So all of these studies, and again, I'm going to the last part, which is what I want to spend most time on. I'm sorry I went a little fast in the beginning, but I wanted to get to the main paper that we just published. But hopefully you see the background of why we're doing this research. Light seems to be working in different cell types and using different mechanisms, but how does it know how to do that? How does light know how to use which mechanism in which cell type? So that's the central part of this paper. Now I'll go a little slowly if you don't mind. I want to actually show you this paper very carefully. So this is the new paper that came out. I'm happy to send this to you, but you can also take the QR code, and this is available online at this in this journal Cells. We had a very good experience, by the way, with this specific journal. I know people have different experiences with different journals, but this was actually one of our good experiences. So this work took about almost, I think, 8 to 9 years. But this was a really, really good piece of work. And I'm going to only show you about 4 experiments in this, but I'd like to go slowly and explain why we did this experiment. So the first experiment we did was, we know that there are different mechanisms. We know that each cell type will respond differently. So in this set of studies, we took the keratinocyte, we took the epithelial cell, we added 2 different scavengers. We added catalase, which is largely outside the cell, and we added N-acetylcysteine, which is both inside and outside the cell. To measure ROS, we also used 2 different reagents. We measured extracellular ROS with a dye called isoluminol. And when you do this experiment, you do photobiomodulation, isoluminol will detect extracellular ROS, right? So the first point I want to make is that extracellular ROS can be detected, which means that PBM is capable of generating ROS. However, if you neutralize it with catalase as well as N-acetylcysteine, both of them will scavenge it and neutralize it. So photobiomodulation generated ROS clearly can be present outside the cell. And again, remember, these experiments are done within 15 minutes, not longer, right? 15 to 30 minutes max. So it is instant ROS in the outside of the cell. The next experiment we did, and the same experiment, but we did another set where we did the exact same N-acetylcysteine and catalase experiment, incubation. But in this case, we use luminol, which goes inside and outside the cell. So we are now detecting intracellular and extracellular ROS. As you can expect, when we measure with luminol, it's obviously at a much, much higher level if you look at the absolute amounts of luciferase. Photobiomodulation clearly is increasing the amount of ROS. Many people have measured this in many different ways. You can use DCFDA, you can use Amplex, you can use many reagents. So you can clearly see that there is intracellular ROS. When you add N-acetylcysteine, which is a global scavenger, it will neutralize it as it did with isoluminol. Interestingly, when you add only catalase, you do not neutralize this benefit, neutralize this generation, showing us that the ROS that is being generated by PBM specifically comes from inside the cell. Make sense? We are using these reagents to tease apart where the ROS is coming from. And by doing this study, we can show that the ROS is actually coming only from the inside the cell, which makes complete sense from everything we understand about cytochrome C oxidase and photobiomodulation. Okay, so then I showed you all this data about ATF4 being very, very important to integrate all these responses, right? So I showed you this data before that if you increase the amount of photobiomodulation treatment, you can increase the amount of ATF4 expression to a particular level, beyond which it will start killing the cell. That is too much. That's when the ATF4 level goes down. And we know that TGF-beta, which is a growth factor that we are very interested in, can induce ATF4 potently, right? So the experiment we did was we added either TGF-beta or PBM, photobiomodulation, and we did the squenching experiment with N-acetylcysteine and catalase. As you can see in these bars here, specific neutralization of extracellular ROS does not omit this effect. It is only the intracellular ROS that is responsible for neutralizing both TGF-beta to some extent, but definitely with photobiomodulation, it's able to reduce this response. Clearly showing us that it is the intracellular ROS that is responsible for coordinating the ATF4 damage response. Then how is TGF-beta able to crosstalk with this? That was our next question. So to do that experiment, we actually started looking at NF-kappa B signaling. It turns out that TGF-beta signaling and redox-based mitochondrial signaling talk to each other through NF-kappa B. Because when you neutralize it with this inhibitor, BAY inhibitor, all of these responses go away. That's all of these numbers here. You can see that we can robustly increase ATF4 with TGF-beta and PBM treatment. But when you use the BAY inhibitor, which is an NF-kappa B inhibitor, you neutralize all of these benefits. And again, I'm not going to bore you with the details, but we looked at many of the downstream signaling, and we did a lot of arrays, and we looked at all of these gene expression, and we use many other inhibitors. We use ERK inhibitor, P38 inhibitor, JUNK inhibitor, a PI3K inhibitor. But all of that basically, this is just gene expression. If anyone is interested, I'm happy to go into the details. But the point I'm trying to make here is that when you do photobiomodulation treatment, the primary target to generate redox is the mitochondria. That redox response directly feeds into NF-kappa B signaling. However, the redox is capable of diffusing outside the cell and activating TGF-beta. And that induces its own signaling pathway through the MAP kinases as well as canonical SMAD signaling. And all of these responses seems to feed into ATF4. We know this because when we look at very specific genes downstream of this whole signaling cascade, we can identify very, very specific signaling pathways that are coordinated. There are specific genes which are down-regulated, and then there are specific genes which are up-regulated. So all of this tells us that even though you're shining light everywhere, outside the cell, inside the cell, on the cell membrane, the signaling that is induced via the mitochondrial redox as well as extracellular signaling gets coordinated, and then we get a concerted therapeutic gene expression response. And remember, each one of these response is different in every cell type. Endothelial cell is different. This is keratinocyte data. We are looking now at odontoblasts and macrophages and endothelial cells. Each one of them responds very differently. So all of these genes are specific for the lineage-specific responses. And this is why when you do photobiomodulation on a burn wound versus a regular wound or mucositis for that matter, even mucositis you can think about, chemo-induced, radiation-induced, and transplant or immunotherapy-induced, right? we think they present as mucositis or stomatitis, but the underlying pathology is very different. However, we know that photobiomodulation can help them, but do we need to change the dose or should we use the same dose? These kind of experiments are helping us answer those questions. Should you use a different photobiomodulation protocol for mucositis from chemo versus stomatitis from immunotherapy is a key question that we could answer with this kind of studies. Now, I want to end with one final paper that we just published. This has nothing to do with photobiomodulation, but we are very interested in TGF-beta biology. And this is a condition which is very common in India, but I'm sure people in Brazil also have encountered oral submucous fibrosis, right? In India, you have all these betel nut chewing and tobacco wad chewing habits, which results in fibrosis of the oral mucosa cheek, right? And this submucous fibrosis, we have always known that TGF-beta has a role. The question is, what is that particular role? And in this particular studies, we were very interested in trying to understand if TGF-beta, if arecolin, which causes submucous fibrosis, causes cell death, what does it do to the surviving cells, right? And we found that TGF-beta that is exposed, we know that TGF-beta is present in SMF, so submucous fibrosis, but it seems to be reprogramming the mitochondria, and it seems to be working through one very specific gene, TFAM in this particular case. And these are the kind of stress fibers that you see in different mitochondrial reprogramming. So why am I showing you this paper in a PBM talk? These changes that are occurring are downstream of TGF-beta signaling, but upstream of mitochondrial signaling. So when you do photobiomodulation, not only can you increase redox, that is actually activating TGF-beta, but the TGF-beta can feed back and change the mitochondrial response, and therefore you're getting integration. This complexity of the crosstalk at different times is why we think, and I think all of you would agree, photobiomodulation seems to be working in so many diseases, right? If there was not such a complexity, if it was simply light and redox, we could have solved the problem, I think, 60 years ago. The reason that we are still trying to understand which protocol in which clinical context is because of this crosstalk. And this particular studies shows you how the initial mitochondrial redox flux can feed back and change the mitochondrial fission and fusion via very specific gene expression. We were looking at TFAM, which is one of the genes of our interest. And we can say that it reprograms the cell and therefore can change the overall therapeutic response. So obviously, they say that complexity is for grown-ups, and photobiomodulation is so easy to do. Anybody can do it. But the complexity is in the dosing and the biological response, right? And that is why I think there is so much opportunity to work together. I think there is an amazing amount of collaborations that we can do. And of course, I get to present all of these nice work, but these are the hardworking students in the lab. Many pictures here of students over the years. And we have a big group, a global group, actually, about 65 people now. And everyone is welcome if you're ever visiting to please visit our lab and love to talk about collaborations and things we can do together. So I'll end with that and I'll turn over the floor. Is that okay? Cool. So
Very
happy to take
nice.
any questions.
Very nice. Fig, are you here?
I think he's muted. Yeah.
Yeah.
Yes,
Praveen,
I
thank you.
am.
Good. Thank you so much, Praveen, for all your talk and for sharing with us all the research that you have been conducting the last, I think, 20-something years, bringing all the basic, from the basic to the clinical applications. And I think that during these years, we have been having some good answers, and we still have a lot of study to do. But this final talk that you have about the intracellular, extracellular, the when you activate one cell, it's something that we have been studying in other disease, for example, in cancer, how the cancer
Right.
transform the microenvironment. So we can see that using photobiomodulation, we can not only modify the cell behavior, but how the cell behavior can modify all the environment. And this is great to show how this is happening. We have a lot of course, mechanisms involved. You are follow some path, but this is amazing, something that we should do. We should better understand this in vitro, in animal models, and also in clinic. So I'm here to say thank you for the whole hard work that you have been conducting. We have here Professor Marcia that also conduct a lot of studies um, uh, regarding more specifically with epithelial cells and also with pulp cells during the regenerative endodontic field. So we have here in this group in my opinion, the best researchers in the world about in the field of photobiomodulation. So
Yeah.
thank you. I think that we're gonna have a lot of things to do together here in INCT, and it's great that all these researchers are here, listen to you, and uh, we can plan something together in the next month, not
Sure.
years, because we are here to produce this science right now. I think that Alan is wants to say something, and Fig, please, you lead now.
Thank you, Manuela. I'm sorry that I'm in this very noisy background, just walking from the dental school to the hospital. So my apologies for this one. And I just wanted to reinforce your words and to thank Praveen for his hard work and also for hosting so many Brazilians for so long. He definitely impacted our country as well in this sense of the scientific progress over photobiomodulation and related. But Praveen, I have one question regarding your fantastic talk, which is a little bit off topic, but it is still related to the progress of photobiomodulation in translational views. I know that the US has recently progressed towards better coding and better reimbursing methods. And I see these as a direct consequence of your work. I see that mainstream healthcare started to understand the basics, the physics, the cellular mechanisms. And as Manuela said, we have lots of very special people like Professor Marcia Marquez as well working. But you are the leader in the US by far in this sense when it comes, for example, oncology. And please, can you share your thoughts about it? How the coding system and how the reimbursement methods are getting better, are getting more tangible now for ourselves. And thanks again for your special contributions.
Absolutely. Thank you so much, Alan and Manuela, for those kind words. We are following, obviously, I said multiple times during my talk, all the fantastic work that's coming out of Brazil over many years, over decades, to be sure. Marcia's work, of course, we follow very closely. I see Alani is here. I see Fabio is here. So leaders in the field, right? And we follow their work very carefully. And I know many of the names, but I don't know. I've not met many of the other people. But I'm sure we have read your papers and learned from you many things that I think we are using in our research. In terms of your question specifically, Alan, we started by actually building on the systematic reviews and meta-analysis. The mask papers that actually came out first. We were actually using that as, look, there is evidence, start recognizing it. So that led to a all federal meeting. So in the US, we have CMS, Center for Medical Services, as well as the NIH, the National Institute of Health. So we had an all hands meeting in 2012, I think 2012 or 2013. We had an all hands meeting where people discussed, is this evidence real? And do we understand the benefit? Do we understand the mechanisms? May not be everything, but is this real and is it safe, right? So those were the questions that were asked. And that led to many of the acknowledgements that you see, including recognition by the FDA that this is not just sauna light or just light therapy. This is actually photobiomodulation, right? It's a very different kind of treatment. And that was huge because once you have a regulatory agency call something different, then the insurers start looking at it. And again, I can point to all these fantastic papers from Brazil and from Europe, from Renaissance Group, where they showed that you can save, I believe the number is $40,000 to $50,000 for every cancer patient because you can avoid many of the complications or reduce the severity of many complications. So if you look at the sequence of events to why we have an insurance code, and if you want to simulate this, and I'm sure you know that René is actually talking to the French government, trying to get a specific reimbursement code in Europe. And once they do it, I think many other countries will follow. So my suggestion to you is you're already working at the highest level of the government. I mean, this grant is a acknowledgement. Your ICNT grant is an acknowledgement that the policymakers are aware of the potential of the field. But having that kind of a meeting where the policymakers are invited and told about the safety, don't worry about mechanisms. They don't really care about the science and how things work in the molecular level. They want to make sure that whatever they're providing to the public is safe first. Second, who is it going to benefit, right? If it's going to benefit the patient and the clinician, they want to know if it is, useful there. And those are the two things that it is that simple, actually, calling these people to these meetings and discussing among ourselves, why is it safe? How can we use it effectively? That's all it takes. That is, I think, the catalyst to get to the next level.
Thanks very much. Fantastic. Thanks for sharing so much for so long. Thank you very much.
Absolutely.
Thanks, Manuela. Over to you.
I see a few more questions. Yeah.
Hi. Thank you. It was a really nice talk. And I have two questions. We will be really quick. Regarding the TGF-beta action, are you planning to do any kind of screening of gene expression after this exposition and this potential mechanism of action? And secondly, are you planning to work for investigate potential additive effects with regular drugs such as anti-inflammatory or antioxidants?
So, simple answer, yes and yes. Long answer, the challenge is the opportunity, right? So, like I mentioned, every cell type, keratinocyte in mucositis and wound healing, macrophages in inflammatory conditions, odontoblasts in dentine regeneration, each one of them requires gene expression analysis and thorough analysis. So we cannot, I'm sure, no one person can do it. Marcia actually has been very, very kind. She has sent so many talented students to the lab, and we have one. I think Maria is online now. She has sent so many talented students to our lab who are helping us do that kind of analysis. So if you're interested in a particular cell type in a disease, love to work with you, right? We would love to work with you on a application. The other problem, which I think you also face and we also face a lot, is the funding, right? Gene expression analysis, unfortunately, is so expensive, even though it has reduced in cost. If you want to do it well, you have to do enough replicates, you have to do enough inhibitor pathways. And unfortunately, it takes time and money. And this is where I think, again, collaboration is where we will succeed. No single lab is going to get the kind of money that even you got, right? This was a huge group effort that got money. So I think we should strategize how to send applications to the US, to Europe, to Asia, where again, there's a whole lot of patients, and Brazil for sure. So those things I think we can do to move forward. Your question about additive effect and synergistic and inhibitory effects, we have thought a lot about that, and it is still not published, but it won a award at IADRA just now recently 2 weeks ago. There are specific epigenetic factors that integrate even this complex signaling. So what I mean by that is odontoblasts, right? Odontoblasts normally live on hard dentine. That is how they are normally structured. But when you injure the odontoblasts or the dental stem cells in that context, you will actually end up putting them in a soft matrix. Now, simulating that in the lab is pretty straightforward. You can take a hard plastic and you can take any kind of hydrogel and put cells on, and you can simulate the matrix. The PBM response is completely different. in the two contexts. So your question about can we, I don't think the question is can we study PBM in different contexts? Do we need to do more than one thing in a patient to get a therapeutic response? And I think the answer is going to be definitely yes. But we need to understand what we need to do. Do we need to use an additional PI3K inhibitor? Do we want to increase the, I don't know, NF-kappa B signaling or reduce NF-kappa B? There are going to be many, many options. But unless we know what we want to change, it is not going to be, clinically, I'm sure you have heard of many things like let's apply honey on the mucosa, right? And then let's do PBM. Should we do honey first or should we do PBM first? That's a simple question, right? So, and both are very helpful, by the way, because it gives you that, emollient effect on the mucosa. But unless you're treating the underlying healing, you will not get that curcumin. You can think of the same thing. There are so many things we can do. The question is, how do we do a rational combination? And for that, we are going back to the first question. We need to do basic discovery research at the gene level.
Okay, thank you very much. Nice talk.
Praveen, I have one question for you.
Uh-huh.
You brought for the literature or for our dental literature the concept of Einstein. I mentioned to you before that at the beginning I was crazy about this. Oh, how can we do with all of this information? But right now I'm truly excited that probably is the way to better understand what we are delivering to the patients, combining all this information. I would love to hear from you. How is the experience that you have been having with the Einstein with the community, with the students, with the tell me what how we are now in this field, because I'm trying to spread the message. I'm trying to use in the papers. I'm really confident that this thing should be used. But tell me, what do you think?
So I can answer that in two ways. In the lab, it works beautifully because we have done lots of, now we have blue, green, red, yellow, orange, you name it. You can use any wavelength and you can combine them very nicely. This concept is helping us do combinations in a very standard way, right? So in the lab, in the research, it is helping a lot. In the clinical scenario, we thought it is so simple, just add the number and do the calculation. But it is turning out that it is not that simple because it accounts for many of the other variations that we have not focused on. So now when we are doing PBM training programs, we actually have a very specific set of exercises that people appreciate. What is this tissue surface irradiance? What do you mean is tissue surface irradiance? And then how do you do the calculations, right? So you're right. I think, I mean, you have to start somewhere, and we started really, at that point. And Alan did a fantastic, his team actually did a fantastic job with, trying to put it in plain speak and published a really nice paper. But I think we need to go to another level. And we have talked about generating an app or something even more simpler like a video. I know you were interested at one point trying to do that. So I think this is, again, a fantastic collaboration on how to operationalize the new dosimetry, not with the goal of, enforcing a new norm, but making it more standardized so that what works in your hands should work in anybody's hands anywhere in the world.
Maybe it will be great to join forces.
Absolutely.
With the NAUT, WALT, ISLD. Maybe we have to try to do something with this focus. And of course, what we can do to collaborate will be great. So, because I think that we should have something.
Yeah, I was going to make a plug for the joint meeting, which is in October. I would love to invite all of you to please attend, and we can find a time during the meeting or before the meeting or after the meeting. We can just sit down and we can discuss how to operationalize the implementation. Because what I think people don't understand, for a long time, I thought if I publish a good paper, everybody will see it and see the value. Turns out that the policymakers don't read science papers, right? They read, whatever news articles and other things. So even presenting to them, I think, was a huge challenge. But now, as we work with larger and larger communities, it is turning out that we have to have more collaborative workshops, hands-on workshops to explain how we do these things. And again, I think I
Okay.
would fully encourage us
But
to explore that.
But do you think that the Einstein could be the best way to talk with the for a reimbursement to talk about the parameter, the prescription?
That is a very good point, because when you present the evidence, even the masked paper, right? So when we presented the masked paper to our policymakers, they looked at it and said everybody's doing it differently. And some of them are not even available in the US, right? So then they were like, how can you do that? So the inspiration for the Einstein came from that, was that we cannot be all saying that it'll only work in this clinic, it'll only work with this wavelength. We know it works with even LEDs, right? That's a huge discussion whether LEDs are useful. We know it works in the patients, right? So the whole inspiration for that standardization came from that, is that we should be communicating in the same language. And the fact that the field is organizing around that is a great sign. It gives people confidence, right? If you were in charge, let's say you were in charge of a government agency, and you wanted to implement something in the health field, you're not going to implement your friend's protocol or your colleagues or your institute's protocol. You're going to look at all the evidence. And if everything is different, then what do you implement? Right? So I think going towards an harmonized dosing and appreciating that there are more than one mechanism, not one mechanism to do everything, right? It doesn't explain how so many diseases are being treated. Acknowledging that, I think, is showing the maturity of the field. And then I think we will get more buy-in from policymakers, from funding agencies, which is again the parallel field, right? If they think it's worth putting into the health population, they will fund research that will support that.
Praveen, before we finish, I would like to tell you that here in Brazil, we have been having a lot of meetings with in vitro group, in vivo group, and clinical trials group. So we are now starting to open for applications for INCT support some studies. But we're gonna have, and of course you are super invited to participate of the like a committee to discuss the parameters, the dose for to be used in this type of studies that we will conduct here in INCT. Aline is here, Dr. Silvia Nunes also, we talk about this, and also other colleagues. So we want to discuss very well the protocols before we start. So I think that in 1 month, we're going to have some studies selected to receive the support and to be conducted in a multi-center way. So we will need you. And of course, after we're going to have meetings with the international ones to discuss collaborations, how to send the students, how to implement some new policies inside the INCT.
Tava ouvindo Sardinha o quê?
Hello,
Pravão. Hã?
Sandrinha? Sir?
Quero ligar. Já
Sir?
pegou. Comprei pra você, meu amor.
I think that's
Comprei
Sandra.
pra você só. Tem na
Sandra.
padaria.
Oh, sorry. And before we finish, I would like to invite all of you. Next week, we're gonna have an extra meeting, not related with the Biophoto Connect, but we invite Professor Saul Paiva from Minas Gerais University to give a lecture about prims and proms, how to evaluate in clinical trials, the patient's report, the patient, how to evaluate better our patients in our clinical trial. So I think that everybody will love to have this class. Professor Saul is one of the leaders around the globe about this topic. So everybody is invited.
So
And
I just wanted to say
Figgy,
two.
sorry.
So I wanted to say two things before we turn it over to Faye. I think research is absolutely, I think what you're doing for research is amazing. I think putting funding behind the best ideas is the most popular way and the right way to get the right information for a policy change, right? Whatever questions we have about safety, about mechanism, about dosing, if you support the right group of people to do the right research, I think that is the best way to move forward. The other thing, and I think Fig is going to probably talk about this, but I think what we absolutely need is a consolidated way to do education, right?
Yes.
Right now, I think I've invited Alani to one of them. Alan also speaks in some of our courses, not my course, but it's the course that Walt organizes through René Jean. He actually leads some of the courses in India and in Europe. So I think if we can make like a larger forum where we are sharing teaching material, but also interactions, that would actually significantly elevate our level of science. Everyone's doing fantastic work, but they're doing it in silos, right? And that's where I think if we can have some more crosstalk, both in education, research, I think we're doing it now, but the education part still is, I think, very isolated. And there are language barriers. Clearly, there are going to be language barriers. There are going to be like some logistics about travel and all that. But I think now, because of technology, I think we can overcome that very well.
Praveen,
Excellent.
we are here 600 far from Porto Alegre with 300 colleagues to have this kind of educational program. So I think that we can spread this message in this way. Thank you, Figgy. Sorry.
Yeah, thank you very much, Praveen. And I agree with you with this attitude towards educational efforts on biophotonics. And I have just a question. It was very comprehensive, your explanation of how tumor cells and normal cells react differently to photobiomodulation. My question is very simple. Do we need any certain cares if we are, photobiomodulating possible oral squamous cell carcinoma or any other type of cancer when we're working?
That is one of the most common questions people ask us, because if you're going to treat mucositis, especially in head and neck cancer, what happens when you shine light on the tumor? So
exactly,
out of pure liability, because US is very litigation prone. I mean, people sue people for all kind of things. We are very careful about what we say officially in every program and every teaching. Nonetheless, I think even the World Association, North American, everybody agrees that we should not treat the tumor directly. So no direct tumor illumination, and for that matter, no direct fetus or embryo illumination, right? We don't suggest directly treating the fetus or the embryo. However, pregnant women and obviously cancer patients are the most beneficial for PBM, right? And as long as you don't directly treat on the fetus or the tumor, I think it should be done. That's our stand on it right now.
Excellent. Thank you so much for this answer, and thank you for all your speech was marvelous. Thank you very much.
Absolutely. Look forward to working with everyone.
So thank you again, Praveen. We are now here ready to start the new part of our meeting, and we'll be in touch for design good studies together. And you, for sure, is a very important person for our INCT. Thank you so much.
Happy to help in every way. Take care. Bye-bye.